Hi, this is Miss Litton, and this is a very crowded room. I love you too. Is that what you're saying? Okay. Very crowded room. Honors Bio Unit 5 review. I made a PDF of all these questions I have here. And if you look in Google Classroom, you will find all of these.
Thank you.
Just an idea. Also, last night I started whipping together some quizzes. Did you see that? And in Google Classroom, because I could assign it like homework, but I didn't want you to think you had to do it. So I made a whole section in Google Classroom that says asterisk asterisk for review, and that's where you'll find the PDF. This PDF will be in that section. I don't have one for development yet. I started working on it, and then it was getting late, so I'll work on that. So you have some questions on development as well. All right? Okay. First things first. We'll go through these, and then I'll try to remember if there's anything else I want to talk to you about. Okay, starting with Chapter 5, can you diagram a cell cycle with interphase? Which interphase, remember, would have what in it? G1, S, and G2. Would you know what happens in each one of those areas? Okay, I would review that. And then name the parts, what occurs, maybe any kind of controls of those different parts. I would make sure I had that as well. Okay, so when I'm talking cell cycle, I'm talking this, right? And then I'm saying this right here is interphase, and then you would have G1, S, G2. This right here would be, I made myself green. This would be called the M stage, but it has two things in it. It has what? Mitosis and what? Cytokinesis, which I didn't put in there very well. You have prophase. What else? What's next? Metaphase, anaphase, telophase. And then cytokinesis overlaps into telophase. Right? And be familiar with each of those. All right. Next. And then once I've gone through it all, if there's certain things you want me to go over, I'll go over, okay? Then we can focus our time. Could you diagram a cell and the stages of mitosis with a cell that has 3 chromosomes, 4 chromosomes, 5 chromosomes? Could you do that and also do meiosis and know the difference between mitosis and meiosis? Do you want me to do that one with you? Yes? No? Yes? Okay. Let me put an extra page in here. So I'm gonna do 4 chromosomes. Okay, ready? No, I'm gonna do 3 chromosomes. Easier. 2 chromosomes. This right here, I don't know what I'm gonna do. This would be like an interphase, right? The nucleus would be solid, right, during interphase. Then when we move into mitosis, what's the first stage of mitosis? Prophase. What's happening during prophase? Nuclear envelope and the nucleolus is starting to disappear, right? The chromosomes become visible. So if I had chromosomes, here we go. When they appear visible, they appear as doubled, right? What are those? Sister chromatids held together by a centromere. How many chromosomes are in this cell? 2. So I would say my 2n number equals what? 2. Just as a review, point of review, this could have originally come from sperm, and that originally could have come from an egg, right? To form a diploid zygote. You all right with that? Okay. That's not part of prophase. That was just to make sure you understood everything. Okay. Then what stage follows prophase? Metaphase. What do they look like in metaphase? Meet in the middle, right? So I would look like this and like this. Over here, what would I have on either end? Centrioles, and I would have what coming across? Spindle fibers. I could have, what are these around the edges? Asters, right? What stage follows metaphase? I guess I'm doing this in a circle. What's the next stage? Anaphase. What happens during anaphase? They get pulled apart. So it's like this, right? Yeah. So I'm going to get pulled over here and over here. And I'm pulling apart this one. Okay, what's the next stage? telophase accompanied with cytokinesis, and you start to reform, right, your nuclear envelope. And then I would have a black one in each one and a red one in each one. What is that called right there? Cleavage furrow. Good. Okay, questions on that part? All right. Next, can you name and describe 3 different ways meiosis contributes to genetic variety and variation? Let's hear some ways. Crossing over, which occurs when? Prophase 1 of meiosis. Second way? Independent assortment, which occurs when? Metaphase 1 of meiosis 1. When? What's the third one? Fertilization, because you don't know what sperm is going to get together with what egg. And then you can have that variety. All right, good job. Number 7, can you describe the similarities and differences between mitosis and meiosis? Which one makes clones? Which one makes 4 cells? Which one makes 2 cells? Good. Which one is done to a somatic cell? Mitosis. What one is done when a baby is growing up into a child? Mitosis. What happens in your ovaries and your testes? Good. Which one has homologous pairs? Both of them have homologous pairs. But in mitosis, the chromosomes line up single file and separate. In meiosis, you have how many divisions? 2 divisions. The first division separates the homologous pairs, and the second division separates the sister chromatids. Perfect. Okay, which one is used to form sperm and eggs? Good. Which one has variety? Meiosis. Okay, differences between meiosis 1 and meiosis 2. Yes?
So is a homologous pair like 4 chromosomes? And then
Homologous pair is a pair, 2. It's a pair. It's homologous pair. So if I get, but keep in mind, I have 23 pairs in each of my cells. I have chromosome 1 from mom, chromosome 1 from dad, my parental units. Chromosome 2 from mom, chromosome 2 from dad. So I have 23 pairs. So they are a pair no matter what. But what happens during interphase, the S stage of interphase, team 3, the S stage of interphase, then they double. Now these are sister chromatids. This is still the pair. Even if they double, this is still the pair. Because I got a mom and a dad. You separate mom and dad chromosomes during when? Meiosis 1. And you never separated them during mitosis. My arms, my leg, mitosis. That's just a copy of everything I got. My meiosis is getting ready for sex. So you separate the homologous pairs, then you separate the sister chromatids. All right, meiosis 1, meiosis 2. So separating the homologous pairs of meiosis 1, and they're considered haploid at that point, and separating the sister chromatids in meiosis 2. Check with your bio buddy, see if they understand that. You understand the bio buddy? Yeah. Minnesota. Yeah.
That'll probably be this.
Yeah. All right, come back to me. Questions you want to ask me about that? Any part confusing? Okay, now, riddle me this. Why after meiosis 1, listening, why after meiosis 1 are they considered haploid, but they're still haploid after meiosis 2?
It's a haploid of the haploid.
Because in meiosis 1 you separated the what? Homologous pairs. Anytime you don't have your partner, you are considered haploid. But then this, they're considered one chromosome when they're like this. Once we rip them apart, they're considered 2. Okay, so that's why this one goes to this one and this one goes, still haploid, because you don't have your homologous pairs anymore. Okay, cytokinesis in plants and animals. In animals, you already told me this, you form a cleavage furrow. In plants, you build a cell plate and then a wall. Okay, can I keep going? Yeah. Okay. Next, can you describe the similarities and differences between oogenesis and spermatogenesis? Oogenesis, what are you making? Eggs. And spermatogenesis, you're making sperm. Sperm, you know, there's millions of them. Since this review session started, you probably made 5,000, gentlemen. Now, since this review session started, I bet none of us ladies in here made any eggs. Okay? So what happens is in oogenesis, we do unequal, uneven cytokinesis so we can keep a lot of cytoplasm in one. And then the other things we make are called what? Polar bodies. And we do that again, unequal cell division, so that way we have all the nutrients in one. All right? Spermatogenesis, you make a bunch of sperm. Okay, done with chapter 5, chapter 22. Okay, can you name the structures of fertilization? Who's involved in fertilization? Sperm and an egg, come on.
Yeah.
I don't know. During special hugs. Okay, what do you call that cap on the top of sperm? Acrosome. What's inside of it? Enzymes. To do what? Digest the zona pellucida, the jelly coat on the egg, right? As soon as the egg and cell membrane meet, you have a quick, right? Almost like an action potential of a neuron. Immediately shuts off so no other sperm can penetrate, and then you have cortical cells that help and make sure to seal it off because you don't want any other sperm coming in. All right? Sorry. Those are the structures of fertilization. Now, can you name and describe all the stages of an embryo in development? So, let's name them. What's first? Zygote. That's right when the sperm fertilizes the egg, right? What's next? What? Marula. What is a marula? Solid ball. Solid ball of cells.
Yeah.
Okay, what's next? What's a blastula? Hollow ball. Okay. What's the cavity inside of that called? Blastocoel. Blastocoel. Blastocoel.
We got that.
Okay. What's after blastula?
Gastrula.
Good. Gastrula is? So it goes solid. Guys, you're too loud over there. Solid, then what? Hollow, then layered. Right?
Yeah.
That layered ball of cells, at first there's just ectoderm and endoderm. Then there's ectoderm, mesoderm, endoderm. Right? And when you have that layered, let me do a better picture. When you have that layered ball of cells, what do you call that new cavity? Right. And what do you call the opening? What does that become for you? Yeah, for us it's the anus, for others it's the mouth. Okay? Then the blastocele right here got smushed away. Right? Then when you have 3 layers, the innermost layer, the endoderm, right? The endoderm, what organ systems are associated with the endoderm? Digestive and respiratory. Think about in your mouth, what goes in there? Food and air, right? Digestive and respiratory system. The ectoderm, what is that associated with? Skin. Nervous system, skin, and eyes. And mesoderm is everything else. What follows the gastrula stage? Narula. Narula. Sorry. This is when you form your spinal cord, your nervous system. Okay? That's when you form your nervous system, your spinal cord. Okay. What day? How long does it take to get to the uterus? 5. Remember we said blastula happens on the 5th day. Okay. All right. On a Tuesday. I had sex last night, Miss Lynn. What were you thinking? Okay. I'm sorry. It's totally inappropriate. Okay. Okay. Any questions about this? All right. Can you name and describe the layers of the gastrule and what each will become? Did we already do that?
Yeah.
Yes, we did. Now, can you differentiate and give an example of an induction and morphogenesis? Ooh. Induction is like peer pressure. One tissue influences another tissue to become something. That's induction. Morphogenesis is the moving around to become that. Induction is the influence. Like the notochord induces the spinal cord to form during the neurula stage. The moving around of cells is morphogenesis, the beginning of that movement, of those structures. Okay, what's next here? Oh, can you describe how mitosis and meiosis contribute to the stages of fertilization and development? What makes the gametes? Meiosis. Once you form a zygote, what's it going to do from then on out? Mitosis. It's going to keep doing mitosis, mitosis, mitosis until you get to the point where you're ready to go, right? And then you will do what? Meiosis. But all the development, what we think of as development, is all mitosis. Next, chapter 20. Yes.
What was the stage where it's growing but it's not getting bigger in size? It's just
Anybody remember that? That solid ball of cells, the morula, cleavage. Oh yeah, I could have said that. That would have been smart. Thank you. To form this morula, you do cleavage. Thank you. Thank you very much. Okay, chapter 23. I'm going to tell you, almost all of chapter 23 are not like straight up questions other than like, what did Mendel work with? Peas. Why was that so slow? Alligators. Okay, most of them are just problems, right? Just doing genetics problems. Do this Punnett square. Cross two curly-haired people. Cross sex-linked traits. Do a blood type one. They're all just those practice problems. So first of all, can you name Mendel's 3 laws and describe them as well as provide an example of each? Let's do it. Law of dominance. If you have a heterozygote with somebody who has a brown-haired gene and a blonde-haired gene, which trait is dominant? Brown. Okay, perfect. What's the next law? Law of segregation. Who is getting segregated? Homologous pairs and then sister chromatids, right? You're separating alleles, right? You always have 2 alleles for every trait, except guys on your 23rd pair, right? Everybody else has 2 alleles for every trait. You're going to separate little t from little t, big T from little t, big R from little r. You're always going to separate those. If that doesn't happen, that's a problem. Law of segregation basically says you do anaphase 1 and anaphase 2. So there's no gamete that's going to have a big R, little r in the gamete. You could have a big R with a little t, but it's not going to have 2 R's in it. You're not going to send any homologous pairs into the gamete. What's the third law? Law of independent assortment, which means that when I have homologous pairs and they're lined up, you guys are driving me nuts, it's got to stop. When I have homologous pairs, they can line up any which way they want to during what? Metaphase 1. So if I line up like this, these go over here. This could be a big R and a big T. Maybe over here is a little R and a little t. But if I went this way, I could get a big R and little t's. I can't do it. And over here, you know what I'm talking about. So I can get any combination, big R, big T, big R, little t, little r, big T, right? Well, now, let's talk exceptions. When are there exceptions to the law of independent assortment? When the genes are linked. If the R and T are on the same finger, they're traveling together, right? Does that make sense to you? Okay? And so, what's the only way you can break up linked genes? Crossing over. Exactly. Okay, and so there are exceptions to the law of dominance too. What are some exceptions to the law of dominance? Codominance. Give me an example of where you see codominance. Okay. Yeah. Your best AB blood type is your best example for codominance. AB blood type. That's what I would go with. Okay. What's another exception to the law of dominance? Incomplete dominance. What would I give for that one? Wavy hair, right? Cross curly haired with a straight hair, you get a wavy hair. Cross a red flower, white flower, you get pink. Not all flowers, right? Some flowers. Okay. Are there any other exceptions to law of dominance? Polygenic traits, where in order to get like skin color, it's not just a simple dominance. You've got A's, B's, and C's all working collaboratively to give you skin color or intelligence. Right? Those are polygenic traits. Sex-linked traits could be exceptions too. Right? Because you guys, you get one shot at your X. It doesn't matter if it's a dominant trait or recessive trait, you're getting it. Whereas for females, we have 2 shots at our X, so regular dominance works. But for guys, it does not. Okay. Can you differentiate between a somatic cell and a gamete? Give me examples of gametes. Sperm and egg. Everything else is a what? Somatic cell. My thing keeps flashing, so I hope it's recording. Looks like it is. Okay. Phenotype and genotype. Phenotype is what it looks like. Okay. Genotype are the actual pair of alleles. You will always have homologous pairs in alleles. You could have 3 homologous pairs, but you always have homologous pairs. A big T with a little t, or a big T with big T, a big T with, I already said a little t. Okay, but you could have 2 T's, 2 R's, 2 Q's. As long as you have all your homologous pairs, that's a genotype. That makes up an individual. Phenotype is what that individual looks like. Homozygous, heterozygous. What's big T, little t? Heterozygous. Big T, big T. Homozygous, dominant. Little t, little t. Homozygous, recessive. Good. Blood types and multiple alleles. So we already talked about that. What are the different alleles you could have? When we say we have multiple alleles, what are those multiple alleles? Big IA. Yes, those are the multiple alleles. We can get any combination of those for genotypes and phenotypes. Incomplete dominance. What was the example you gave me for incomplete? Pink flowers. Good. Curly hair. Those are the ones that are expressing the incompleteness, right? The red flower and the white flower aren't. Straight hair and the curly hair aren't. The pink and the wavy, the one that looks in between. Sex-linked traits. Those are another way we could say sex-linked for our benefit is just what? X-linked. Right? Just X-linked because it's on the X. This right here could be normal vision female, yes? X big N, X little n would be what? Carrier female. What is this? X big N, Y. Normal male. What's this? Colorblind male. Got it? Okay. All right. I put N's there instead of C's because it's easier, or instead of B's because it's easier. Think normal. Yeah.
Would it be possible to have a carrier male?
Is it possible to have a carrier male? No, you either have it or you don't. When they talk about carrier, you're usually carrying it, hiding it. You don't see it. Okay, what's an example of codominance? AB blood type. Roan cattle. Like 2 different colors of cattle, and you see both colors in the cattle. Like the black bunny, white bunny, you have black spots and white spots, black hairs and white hairs. Okay? Polygenic. What's an example of polygenic? Skin. Intelligence, right? Good job. Okay, next. Can you identify and give specific examples of multiple alleles, incomplete, sex-linked, codominance, and polygenic inheritance? Yes, we just did that. Can you do a Punnett squares with 1 trait and give genotypic and phenotypic results with 2 traits? Remember how we do the product law? Do individual Punnets, multiply them together. Can you do explain? Can you do explain? Yes. Why a test cross is necessary? Can you do it? Explain it? Yes. Why would I need to do a test cross?
The likelihood?
No? I don't know the genotype of a dominant phenotype. He has brown hair. I do not know if he's brown or brown blonde. Right? Somebody who has blonde hair naturally, not me. I know what she is. I know what you are. I know what you did last summer. Okay? Then, I know you. I know you. Okay? I don't need to test them. They're a recessive trait. Okay? The brown-haired people, the brown-eyed, the dominant phenotype, if that's expressed, if you have a tall pea plant, who are you going to cross it with? Short. Right? Because if they're a carrier for the short gene, you'll see it at least half the time, right? Okay. All right. Next. Do the practice problems in the chapter, and at the end of the chapter, and at the answers are in the back of the book. And do your additional objectives. Okay? Questions or concerns that you might have that you would like to discuss with moi? Yes?
Can you give an example of morphogenesis?
Oh, yeah. Morphogenesis would be like when your eyes move into a certain place. Morphogenesis is when you form a gastrula and you fold, the tissues fold. That's all morphogenesis. What else? Yes?
So if you had big R, little r, big R, big R.
Wait, let me write this. Let me get a fresh page here. Big R, what'd you say?
Big R, little r, big R, big R, and little r, little r. Are those all homologous pairs?
Yes. This would be on a homologous pair of chromosomes. This could be on a homologous pair of chromosomes. This could be on a homologous pair. And during interphase, they would replicate, right? Sorry, I'm getting too big. But you understand what I'm saying? Okay. Anything else? Yes?
If we have to solve anything on the test or do like a Punnett square, are we allowed to have like practice?
The whole, if it's on the multiple choice part, it'll be on the back of your Scantron. That you'll have all the backside to work on whatever you want. Yes?
So, let's say an example. So let's just say the big R and the big R. So those would be a homologous pair, and like a pair of a chromosome and a pair of a chromosome would be homologous to each other.
Well, yeah. I don't know why I can't write. Let me try again. Oh, good. This, okay, if that was a gene on a chromosome, that's a homologous pair of chromosomes. Big R, big R. Okay, now ask me a question.
So that would be a homologous pair, and if you had another set, those would be homologous to each other.
These right here are sister chromatids. This is still a homologous pair with this over here. These are sisters and these are sisters. This is still a homologous pair, whether it's double or single, it's still a homologous pair. Because this one right here, let's say this one came from mom and this one came from dad. That's the homologous pair. Yes?
So basically from one homologous pair, like it ends up becoming 4 chromosomes. Meiosis, right? Like
Not
at the end.
ultimately.
Yeah.
Yeah, it gives, yeah, because you're going to do interphase and that's when they're each going to double. Okay, what else? Yes?
Can we do a Punnett practice problem?
Sure. Like this? And I want to know how many will turn out like this. I want to know how many will turn out like this. Write it down and then look away because I'm going to solve it. Do you have it? Big R, big T, big S, little s. Heterozygotes all the way across. With little r, big T, little s, little s. Okay, do not serve it, don't solve it out loud where everybody can hear you or even your neighbor can hear you because it's annoying. Everybody wants a chance to do it, so don't do it out loud, okay? Be careful with things like S's because they look the same, big and little, and W's. Okay, so you can check your work up here. Want another one? What? Yeah, I'm gonna give you another one. Yeah?
So what does it mean when it would, let's say, give you those? Thank you. Like, it would give you that, and then it would be like, what are the possible gametes? Is that just what are the possible?
These,
like,
the gametes are on the edge. These are gametes on the edge.
So like when it would tell you.
I wouldn't ask for possible gametes from each of these. I would have it something only when there's two, and you could just foil it. Yeah. Okay, riddle me this. Baby Jane is type O blood. Mom is type A blood. And two potential fathers, Jack and John. Don't say anything out loud. Okay? Jack is type B blood, and John is type AB blood. Now, you may be able to guess right away who's your daddy. That's assuming mom only slept with Jack and John. We don't really know. But also give evidence to why that would be. Okay, give evidence as to why that would be. The first thing I would do is start off by writing everybody's potential genotypes. That's the first thing I would do. Is that what you're doing? Yeah. Okay, so once you have this written down, I'm going to start solving it here in the background. Okay, so don't look if you don't, and please don't work it out loud again where everybody hears you because it's disruptive to those who are trying to think it through. You're solving it out loud, which is exactly what I meant to do.
I was explaining it to your question.
Does that make sense to you what I did? Mom, well, Jack's buying the diapers. Whether they're in love, I don't know. Okay, but Jack is buying the diapers. Okay, do you want another one? Yeah. Remember, I'm recording this. Lord willing, it goes up. No problem. Okay. Jane. Jane's father was colorblind. Jane's father was colorblind, and she had a boy and a girl. And tell me if Jane married a normal male, what could be the condition of all of her children? Jane's father's colorblind, and she married a normal male. She had a boy and a girl. What could be the condition of all her children? Okay, so I'm going to start to work out genotypes. Okay, Jane's father is colorblind, so I know she has to have at least what? One bad X, right? One bad X. So I didn't tell you Jane had normal vision, but she does. I should have included that. Jane has normal vision. you get a chance to work that out? So if I asked you about her children, what are the odds of her children being colorblind? What would you tell me? 25% of her children could be colorblind. What if I asked you about her girls? 0. But what could they be? They could be carriers, but they're definitely not colorblind. Okay, you know what I'm saying. What if I asked about her boys? 50%. Because I would predict half of her boys would be colorblind. Okay, I got a meeting. I love you. Watch the review. Make good choices. Have a piece of toast. Tell them goodbye. Bye. That was weak. That was really good. Tell them goodbye again.
Bye.
All right.
Study.
Study.